given these single chromosomes floating around the nucleus, you'll notice that they're always in even numbers. We have 46, and then in many other creatures, you'll find many different total numbers floating around. But here's the interesting point. Whenever a biologist says humans have 46 chromosomes, or they mention that other creatures have, for example, the fruit fly has 6, stop just for a moment. They'll then say something like 23 pairs. If some species has 40 chromosomes, they'll stop and say, 20 pairs. What are they talking about? Why do we do that? And the answer is that a given chromosome, and watch my finger float around the nucleus, there's this other chromosome floating around the nucleus, not touching, and it has the same genes running down its length as the other one. An individual gene in question, which has a place on this chromosome and a place on that chromosome, may have different instructions, different particular developmental details that it influences. But it influences that step of development, that role in the creature's development, the same way as on the other chromosome. So this is interesting. Every gene is really two locations, two separate, not touching places in the nucleus of the cell. And this can be a little misleading. You've heard about big A and little a for the two different instructions, and the way we write them, they look like they're attached together, but they're not. And they have, again, nothing to do with the attached replicated chromosomes during the process of cell division. No, we have, think about in your cell, one of your cells, here's its nucleus, and inside these 46 chromosomes floating around, and yet, in genetic terms, 23 pairs.